Okay, so we're going to look at DNA today. DNA is probably one of the most important molecules in biology. It contains instructions which code for every living thing on the planet, but it's probably only in the past 50 odd years that we've begun to understand it. So as you should know, it stands for deoxyribonucleic acid. Deoxyribonucleic acid. It's called nucleic acid because it essentially is a weak acid. It's found in the nuclei of cells. It's a polymer, and it's composed of lots of things called nucleotides. So a nucleotide then, what you should know is the deoxyribo part stands for the sugar. The ribo is sugar that's in there. It's a 5-carbon compound, and that sits wedged between the base, so sugar, and up here is a phosphate molecule. And that is a nucleotide. Another nucleotide. Phosphate group is negatively charged, and it gives pretty much their acidic properties. Gives the DNA its acidic properties. Your pentose sugar, that's your deoxyribose, and then the base over here defines what the nucleotide is. So the bases you can have that can fit in there: A, T, C, and G. So adenine, thymine, cytosine, and guanine. So you don't need to know those full names really, they're just there for a little bit of extra. So how do these join together? So there's a polymer, so they join together and they essentially form the DNA molecule. So I'll take this one here, so this molecule here, and how does that bond? So what happens is this carbon here will bond to the next phosphate molecule here, and then you'll get your base there, and so on and so forth. Okay, all the way down the line. So this bond here, this one here, this one here, those are also phosphodiester bonds. So phosphodiester bonds, and essentially all that is, when they bond, it's a condensation reaction and water comes off. 2 nucleotides forming a dinucleotide, 3 trinucleotide, and then a few form what's called an oligonucleotide. It's a few of them, and then many of them as a polynucleotide. DNA then, it was discovered in I think it was 1953 by Watson and Crick. Okay. They were at Cambridge, and they used some data generated by a couple of other people, a guy called Wilkins and Rosalind Franklin as well, and they took this forward and they won the Nobel Prize with the structure. They essentially found out that it was double-stranded. It was double-stranded, and they run anti-parallel to each other, so in opposite directions. It's double helix in that it winds around each other. The bases, so the strands are joined by hydrogen bond. Okay, so if we look up here, so that will be another base there, and that would be in there. That'll be a H bond, and the base pairing is specific. So A pairs with T, C pairs with G. Okay, so that's the structure of DNA. We'll do a little bit more in the next video, and then we'll look at how it replicates.